pegylated species. So my role is typically, whereas Daniel is the CMO, my role is normally as a consultant to a small virtual company with my mission to get the drug approved. So I work as an interface between the CMO and a regulatory agency. So I'm usually the one that has to write these justifications for why there should be 10% of something instead of 1%. So what I'm going to do today, I only have 22 slides. I'm going to go through a number of examples of peptides, drug substance, drug product, and then I'm going to switch over to how do you set qualification levels and how do you justify that. So in the world of conjugates, your life is a little more complicated because you have extra specifications. The universally accepted paradigm is you have a specification of your key intermediate, which is your pre-conjugate molecule. It's typically a TFA salt of a peptide for this audience. It might be a oligonucleotide or something like that too. You set a specification for that. It's released, and I'll show you an example of that. You do the conjugation reaction, and then that is your drug substance. That, of course, has its own specific specification. It's more elaborate than a normal peptide because you have all the ambiguities associated with whatever you conjugate it to. And then you have your drug product. In addition, whereas Bachem is used to seeing 111 specification, as an end user, I see these things, see these doubled up. So I may have a specification for Bachem, and I may have a different specification to the FDA. So one's a regulatory spec, one's a CMO spec. CMO, you always hold to the tightest requirements, and if they complain, you throw them a little bit more money and you say this is what you have to meet. But as a part of a company, you want your regulatory spec to be as loose as possible. You don't want a lot to fail. In addition, in the United States, you have a single drug product spec, a single drug substance spec that reflects end of shelf life. You may have an in-house spec that says, I want it to be 97% when it was first made, and I'm going to let it degrade down to 93%. And only the 93 is what you file to the regulatory agency. In Europe, it's a little bit different. In Europe, you file for the drug product a release spec and a shelf life spec. So when I use generic specs here, I'll show you over the next half a dozen slides or so. You have to remember these are sort of what I'm envisioning as a regulatory spec. So this is the sort of things that regulatory agencies want to see. And I'm just showing you examples. So this is a garden variety TFA salt of a peptide. There's nothing abnormal about that. You're going to do an identity test. You almost always do mass spec, retention time. You're going to do AAA or CHNN or something like that, or sometimes both. Do AAA to determine that the amino acids are in the correct proportions. You almost always now do a combustion analysis to do a total peptide content. And you'll see how that shows up in later tests. But there's nothing unusual about that. It's pretty simple. Early stages, of course, you try not to set numerical specifications. You try to get away with reports. And most of the time you get away with that. Later on, you'll have to hone in and put numbers on things. When you begin talking about a drug substance, and particularly conjugates, then you have to be thinking like the FDA. In particular, if you're conjugating, for example, PEG to your molecule, they and everyone knows PEG does not absorb. You're not going to see it by UV detection and reverse phase. They want to know how much is in there. So you almost invariably have to run a test to determine the excess amount of your conjugate. It's not so hard if you're just conjugating a dye or something else, but like a fatty acid, you would have to determine how much was in there. Your identity tests tend to shift. You'll see this on the next couple of slides. Particularly for large molecular weight PEG, you can get a molecular weight by MALDI, but that's not sufficient for an identity test. You still do the test, you just put it somewhere else on the specification. If your molecule is complex, you will be asked to do a bioassay. Nobody wants to do a bioassay. Everyone assumes that their compound can be adequately characterized. Your best bet is to set up some bioassay that is simple, like a binding, a lysa binding. Find an antibody to your compound. Find an antibody to your PEG. It's almost impossible to get out of doing a bioassay. And one of the other things here that is the force degradation, because the PEG tends to dominate and other dyes and things like that tend to dominate the chromatography, your best bet is to do a lot of upfront force degradation of acid, base, light, heat, oxidation to really map out and verify that your method is going to detect any possible change. The worst thing in the world is to discover after the fact, 2 years later, you've had some sort of degradation that you did not uncover in your original method, and then you have to come back and reanalyze and deal with the ambiguity of how much the material changed over time. So do a lot of force degradation up front if you're dealing with a conjugate. Here's what an early drug substance specification may look like. I'll point out a couple of things. In red here I've added, if you look at this for a second, you can see it's pretty much standard, reverse phase, ID. You're going to do your molecular weight by MALDI, but it's no longer going to be called an ID test. It's going to be called just physical chemical. And that's because the FDA knows that it's not very discriminatory when you have a large molecular weight. They want to know what the molecular weight is, but it's not going to distinguish between peptides of related sequence. You're going to do, in this particular case, it's almost all UV, reverse phase UV. In this case, we've been successful in doing this, a total impurity summation of perhaps one or more methods, but only based on UV, and just have a residual PEG by, in this case, a charged aerosol detector. So the FDA really wants to see that. I mean, I just filed an IND 2 months ago, and we did not have that in there. And we alluded to the fact that we were developing that, and the first response was, we want to see that on the next lot. So they cut us some slack, but they said they want to see that. The other thing you see early on peptides, particularly conjugates, is a nonspecific content method. Of course, you have to determine the content of your material. You're no longer allowed to count the water and the salt as part of the active. That went away a couple of years ago. In the beginning, you often don't have a reference standard. You don't have a quantitative reference standard. So what you can do is use AAA or combustion analysis to determine a total peptide, and you can normalize that back to a pseudo-specific test by purity. And a common thing is, for example, Daniel said a peptide content of greater than 85%. Multiply that by your 95% purity, and you'll get your, whatever that math is, 88%. And you report that as a content. You'll see that that actually changes over time. But for an early product, something like this might be very typical. Microbial endotoxins are very standard levels. And what I tried to do in here is I put some numbers that are pretty routine. They are product specific, and it's almost impossible to come up with examples without getting caught up in the details. I'm just giving you the highlights. And elemental impurities and all these are all specified by whatever your route is and things like that. Oops. So as you go in the substance from early to late, I touched upon some of this. Your tests get more advanced, your methods get tighter, you tend to put numbers on things. It's always the best bet not to put a number on something if you cannot scientifically justify it. So it's the easiest thing in the world to say, I want less than 10% TFA in my product. That's logical, but then when the FDA says, why 10? Why not 9? The real explanation is I made up that number. But that doesn't go very well. So I've always been taught, don't put a number unless you can justify it. Get away with report or wide ranges. When you can begin putting tight justification is when you have real numbers. So something that do change, very typical I found is a single ID test is sufficient, but then you have to move into following Q6A, which says 2 identification tests. Sometimes you can get away with a reverse phase with a UV scan on it and say that's specific. More often than not, it's 2 different chromatographic techniques. What I talked about earlier is the assay content. So again, you can use a non-specific normalized by purity. Eventually, you will need a representative reference material that you're using by HPLC area counts, normalizing against a weight standard. And that'll be the gold standard. As you develop more batch history, some of those initial tests, for example, free PEG, if you make 10 lots and you demonstrate that it's never been in there, perhaps you validated the assay, At that point, you can easily justify and say this test is no longer really needed for specification. And I should have pointed this out earlier, but specifications are things you have to do every single time, and you have to do a subset of those on stability. It's a burden to have to do a bunch of tests. The more you can slip over into characterization one-off tests, the better you are. You don't want to have your CMO have to do some ridiculous test every single lot when you know in your heart it actually doesn't provide any information. So one quirk that I've noticed in the last years, in particular, I fight Bachem over this all the time. So I have a number of clients that work with Bachem. And Bachem's QC group says, we will not identify acetate as the counterion, even though it's done as a standard in the test. And I keep pointing this out to them. If you look at Q6A says if your material's a salt, you need to identify the identity of the salt. They don't listen to me, apparently. So as the interface on the small company, we put identity of acetate on the C of A. So we take the Bachem C of A or the other manufacturers. We add that to the regulatory spec, and we rely on our information, knowing how the CMO runs the test, that they do in fact use an acetate as a standard. It's typically an HPLC based, and yes, inherently it is an identity test done. So we can justify that, and if we really had to, we would force the CMO to put that on the C of A up front. But that's my little dig, because I know Matteo has always said, no, you don't need to do that. Here's what something in a late stage may look like. It's, you can see why I've sort of morphed it here a little bit. I've added a second identity test. I added the identity of acetate. And you have to realize some of these identity of acetate and acetate level, you'll see on the next slide, it's the same test. That's not hard to do. What else did I do? I think I added also a different type of impurity summation. I've added a second impurity method here. So I've added both impurities by reverse phase and SEC, and I've added examples of how you may begin to specify those impurities. At this specification, they're just tracked by retention time, which is a step in the right direction. Ultimately, if they're on your final spec, you would have an identification of them. But at this point, it's implied that I've begun tracking them. I have a reporting threshold that's low enough. I follow them by retention time. And after the process has been validated and I've seen everything and I know what the commercial product looks like, at that point I'll decide what the final structure should be and actually assign them a name. So you do get a little bit of leeway before you have to identify stuff. You should provide some partial identification, typically a mass spec, and perhaps a matching to a putative degradant or a process impurity. And only at the final end, when you've actually validated the process, will you go to the trouble typically to make the impurity and spike it in and show what it is. The second half of the late stage, you see what I've put here is oh, that's one too many. I've added a specific ABC contents now reverse phase. So this is against an external standard. And I have the bioassay here. So if you look at drug product spec, this is for a hypothetical lyophilized powder. So spec looks as much as you, sort of typical. I've got an early stage. I have a single impurity. I'm going to need two eventually. I have a purity by HPLC and impurity profile. It's the same analytical method. What's important here is early on you track total impurities, because typically you have not identified whether they're process impurities or degradants. There is no such thing as total impurities in a drug product that's commercial. It's total degradants. So in the beginning, you capture everything, probably have an impurity value that's lower. When you go to commercial, your impurity value is going to go up because you're only going to be dinged for the degradants. That's the only thing that's going to count against you. So you'll see that in the next slide. Hypothetical lyophilized powder. I mean, if this was a liquid, you would have content in a volume in container. This is a lyophilized powder. You can do content uniformity, which takes anywhere from 10 to 30 vials. If you look at the guidance closely, that guidance applies to commercial products. The development products, they have a clause in there that says you can use an in-process control. So in this particular case, IPC, in-process control, if you'd done a fill weight during the fill manufacture where you had rejects of plus or minus 5%, as indicated here, you can pull that forward, put that on the C of A, and call that content uniformity. So you don't have to cannibalize your precious drug product vials until later when you're at full scale, when it's not a big deal to sacrifice that many. I always hit these buttons wrong. So early, late stage, the other thing, let me see what else I put here. The other important, so your second ID is going to come up. You've also never validated your analytical methods for the impurities until the very late stage. So there's some ambiguity as when you report them as area percent. You don't know the relative response factor. You don't know if the LOQ, LOD has been established. So there's some ambiguity, and you can take advantage of that when in the next portion of this talk when you see the qualification levels. But just remember, what's looming forward, if you haven't gotten there yet, you will be making all the impurities as standards. You'll be spiking them in. You'll be revalidating the method with those standards to determine the LOQ, LOD for each of the specified impurities. So while early on you may have been done a method validation where you did, use the parent species, watched it disappear, set an LOQ, you're not done yet. You've got another big round of validation coming up in phase 3. So here's what a typical late stage drug product may look like. What's notable here is I've switched from impurities to degradants. So that means when you have specified degradants, by definition at this stage, well, I'm implying that this is very close to NDA. It's not clear from what I've put here, but these are all degradants. The process impurities were captured at the drug substance stage, which is why your total degradants are here. And you'll see some of the numbers don't, you can have a lot. This is end of shelf life. You can have 10% degradation, right? And purity of 90% is typical. The other thing I'll heads up, this I can tell is a hypothetical clinical stage because there is no such thing of purity for a drug product for commercial. That just goes away. If you look at the guidance, the purity is not mentioned for a drug product. That's only a clinical stage. In the end, a drug product is just how much did you give, and the sum of the impurities defines what the purity is. So let me switch gears a little bit because I find this more interesting because this is, and I framed it as 3 different slides with hypotheticals. And so this is what I really love to do. Spend a lot of time doing this. So just as background, Q3A, you can read this, everyone's probably read it. It's a big, long, lengthy thing. It says you must qualify. Let me cut to the chase because there's 2 sentences in this guidance that you need to pay attention to. One is, any level of impurity present in new drug substance adequately tested and or in safety and or clinic. That is a killer. Is it and? You need stuff both in animals and clinical, or is it an or? And it's at the reviewer's discretion. So there's a worst case and a best case. I can tell you now the trend in Europe is it's and. You can have high levels in tox lots and in animals. If you haven't put those impurities into people, good luck, you're not getting them. So the take-home message here is you need to get the stuff into people. The other one is, what's always clear is your level in a tox lot isn't actually related to how much you're going to end up with. It's loosely correlated, but it's not. If you have 0.2 in a tox lot, your qualification level isn't 0.2. You have to do more complex calculations, and I'll show you that in the next couple of slides. So just a reminder, Q3A is substance, Q3B is product. Nobody knows Q3B. The take-home message is the levels are a little bit higher. The other thing about Q3A is, first page, it doesn't apply to peptides. It does not apply to compounds in the clinic. Q3A and Q3B applies to commercial products. And sometimes the regulators are upfront with that. I mean, I met with the FDA last week, and they said, before the meeting, they said, please follow Q3A. And we made a big song and dance and cried and said, we can't do that, this is as good as we can do. And they said, well, you know this doesn't apply to the clinic. What you propose is just fine. So some of it's putting a little fear into you, I think, to make you work a little harder. But part of the things I'll show you in the next slide, the FDA has published human equivalent dosing conversion, so you can look up these numbers. But here's a typical scenario. They bring me in as a consultant and they say, FDA told us to do something, what do we do? So you have a product that, a particular impurity, you don't know what it is. I mean, it's a peptide probably, presumably. It's in the tox law at 0.58. You know what the no observed effect, adverse event from the clinical. You know what the clinical wants to dose. What you don't want to do is just go to your, the simplest thing is to put the limit at 0.58, but do you really need to do that? Do you want to limit your CMO to not have more than 0.58 at that? So what you have to do is you have to do the math. So in this particular example, 0.58%, you dose it out to the mg per kg in the whatever tox species, you convert it to humans, you can see the math there. And then you use, in this case, this is the most conservative because I'm using the largest anticipated clinical dose, setting the worst case scenario. You follow that out, and what was in the animals at 0.58, if you assume an exposure multiple of 1, you can have as much as 2.4. So with a straight face, you could you could work with the clinical people, tox people, you could commit to the agency, we will not have more in our next human dose than we anticipated, than we had in the animals. And that's the sort of number. That's how you can go from a 0.58 to a 2.5. Most companies don't take that level of aggression. So you may want to, tone this down and say, 1.5 has a good, feel-good number. It depends on your interaction with the CMO and what's possible. Here's another case where, this one is clearly a degradant. It's in a tox lot, drug substance -20, never changed, no big deal. Made the DP and it grew, so it's a degradant. In this case, I've changed the numbers around. You've got the clinical dose, what you anticipate it to be. It's clearly a lower dosing. It's 0.5 mg per kg. So what level do you do? So in this particular case, you do the math. And you would say, just looking at the drug substance, 0.28% times that safety. This was in a monkey, so you divide by 3.1. You come out to the max dose, and you see you could have justified 4.5%. But clearly you're not going to set 4.5% in your drug substance because you know it's going to degrade in the drug product. So you may, in this particular case, look at, it's the highest lot was ever made, 7.77. Let's bump that up to say 0.9, and you can do the math and say, I know it's at 24 months. I'm going to bring it out here to 2.1, and you could easily justify having something that apparently is 0.28 up to 2.1, and to continue to follow it. Right, part of the clinical exercise is to evaluate the safety. So you could justify going that high. So the last example is sort of the gold standard. This is, now it's time for the NDA. So now you've got to compare animal to human to process history. And I'll show you in the next slide, which is the last slide, some actual real numbers. But you can see you've got now manufacturing says, I want drug substance 0.8. Drug product people say, if I'm allowed 0.8 to start, by the end of shelf life, I need it to go up to 1.5. The clinical says our clinical dosing, the max is going to be 0.35. And now you're stuck writing that section of the NDA. How do you convince the tox and the CMC people to allow you to, in your justifications of your spec? And so when you do the numbers, which you have to do the numbers a couple different ways. Now you have to determine the maximum in the species. So you might have 0.28, for example. Well, you have the max allowed in the animal. Now you have to take your proposed specs. Your proposed spec and substance was 0.8 times the max dose. You end up with a number of 0.32 mg per kg compared to the animal dose 0.0028, and you can see you're 11-fold over. So you're at the drug substance limit. Your animal tox exposure multiple is 11-fold, so that's very good. And this last one is, if you take it all the way to the drug product, it's about 6. So if you put that all together in a table, this is what it might look like in an NDA, for example. This is a compilation of tox, clinical, drug substance, drug product, manufacturing, all condensed into a table. So it is possible to make it like this, that you spend a minute looking at that and you go, why isn't this presented like this more often? But it does actually make a lot of sense. And you can see this was that one particular example, I think. 0.39 tox, 0.57 was the highest in the clinic. You've got exposure multiples of over 10. You're doing by multiple species. I showed you this example of 6 in a rat, but in the monkey it was 12. And so you can do things like this, and you can see 0.37 in a tox was justified all the way up to almost a tripling in the drug product. Some things are allowed. Here's 3.6% of an impurity, and actually I can tell you this is an approved product. 8% allowed in the drug product of a degradant. Justified it. We did the tox. It was fine. You can see all the numbers. We have exposure multiples are quite high. So I just want to give you highlights of that. And with that, I think I'll end and we'll go into the discussion.